Good morning, everyone, and thank you for joining in. I'm very honored to be here today to present to you how Polyplus-transfection has been supporting viral vector therapies from process development to commercialization. Polyplus-transfection is a biotech company located in Strasbourg, France. Our products are available worldwide. We are focused on providing a key component for gene and cell therapy, namely transfection region, which play a critical role in the upstream manufacturing of AAV viral vectors. Since the creation of the company about 20 years ago, we were heavily focused on quality. Hence, we were ISO 9001 right from the beginning. We accumulated 20 years of expertise in transfection, leading us to launch the first GMP transfection region available worldwide in 2018 with our PI Pro GMP, which is currently the global leader for transfection region in use in gene therapy. In 2020, we've launched a new product, Vector-V AAV, which is dedicated for AAV manufacturing, especially in suspension cells. Our mission is to provide high quality, reliable and innovative transfection solution, especially for gene therapy. Our primary focus is to continuously innovate for the gene and cell therapy field. As you know, viral vector is the preferred delivery method in the majority of ongoing nucleic acid therapy strategies to efficiently carry a gene of interest into target cells by either direct injection in a patient, namely gene therapy, or by first transducing patient-isolated cells that will be then reinjected into the patient, namely cell therapy. For both strategies, these viral vectors are manufactured in human embryonic kidney cells, HEC293, using transient plasmid transfection. The number of clinical trials using viral vector, such as adeno-associated virus and lentivirus, has been continuously growing over the years. As you can see here on the data shared in the ARM report at the end of 2020, there are over 1,100 clinical trials ongoing worldwide, with a heavy focus on oncology and cancer treatment, leading to increasing needs in viral vector manufacturing. The amount of viral vector needed to treat a single patient is highly dependent on the therapeutic indication, as well as on the route of injection. For example, for ophthalmologic treatments, 1 × 10^12 AAV particles per dose will be sufficient, while a hemophilia treatment will require 1 × 10^15 AAV per dose, hence 1,000 times more. As the number of projects that are moving toward late clinical phase and commercialization are increasing, the quantity of viral vectors needed has also increased. To improve the global production, the productivity of the manufacturing process has to be improved by switching to suspension culture and by optimizing both upstream and downstream manufacturing steps. To reach high viral vector productivity, improvements to the manufacturing process are also required to simplify, standardize industrial processes and reduce the overall global cost of production. In addition to productivity, reaching commercialization for more gene therapy projects has increased the need to better secure the quality of raw material and the supply chain. Hence, it is critical to work in collaboration with trustworthy suppliers to ensure safety and reliability of AAV manufacturing and then gene therapy treatments. The manufacturing of viral vector requires several steps in order to obtain the final release product that is categorized as an advanced therapy medicinal product, or ATMP. The transfection steps occur very early in the upstream process and is critical to ensure the high virus titers needed for gene therapies. Raw materials is a general term used to describe the starting materials and reagents used in the production of a drug. When manufacturing viral vector for therapeutic purposes, the cells, medium, plasmids, and transfection regions are considered as raw material. As you can imagine, the quality of raw materials is key for the success of the manufacturing process and the quality of the final ATMP. At Polyplus-transfection, we are providing two different transfection regions for ATMP manufacturing, PI Pro, and more recently, Vector-Via AV. I will tell you more about this in the next few slides. There are three main challenges of large-scale manufacturing of viral vector. Consistent and reproducible manufacturing of AAV is not simple, as several parameters can vary and impact the viral titers. Scaling up AAV manufacturing also shows some technical challenges, especially when going from adherent to suspension cell culture, and is quite a time-consuming process. Finally, the regulatory requirements for raw materials in ATMP manufacturing have increased drastically lately and need to be addressed early in the clinical development process. Let's walk through those three challenges in the next few slides. With this in mind, we developed Vector-Via AAV transfection reagent and launched it in 2020. Vector-Via AAV is specifically developed for large-scale transfection of suspension cells. It is a cationic molecule allowing efficient compaction and transfer of several plasmids simultaneously in serum-free conditions, hence optimized for AAV manufacturing in HEC-23 cells grown in suspension. Vector-Via AAV allows for increased AAV titer yield. It comes with improved protocol for large-scale transfection. It is very flexible and is compatible with different cell culture systems. It has been developed for large-scale transfection and it will soon be GMP compliant. Let's first focus on how we can generate consistent and reproducible viral titers. Vector-Via AAV transfection reagent has been developed to drastically increase the viral titer yield. Currently, one of the main bottlenecks in gene therapy is the amount of viral vectors that can be manufactured. With Vector-Via AAV, virus titers can be increased up to 10-fold, hence allowing us to produce more viral vectors for gene therapy. Not only does Vector-Via AAV allows for increased viral titers, it also offers excellent reproducibility and scalability. Here you can see that we obtain the same viral titers, whatever the volume of cell culture transfected, over a 50-fold cell culture volume increase and different types of culture vessel. Hence guaranteeing a very reproducible manufacturing process, which is critical for industrial manufacturing. Let's now focus on how we can improve AAV production scale-up, which is currently very technical challenging, especially if you're moving from adherent to suspension culture, and also quite time-consuming. Challenges of industrial AAV manufacturing using transient transfection includes the following pain points. Usually, protocols developed during process development are not very suitable for industrial use. Managing cost is extremely critical in an industrial setting. In terms of the transfection process itself, the volumes to be handled to make transfection complexes can be quite large, leading to some variation, and the transfer time to bioreactors may be long, up to several hours, depending on the volume of the bioreactor. When developing Vector-Via AAV, we thought about addressing all these challenges with a user-friendly protocol adapted to industrial processes with reduced transfection complexion volumes. The transfection complexes are stable over several hours, leaving plenty of transfer time. And finally, by increasing viral titers, we are reducing manufacturing costs and making gene therapy treatments more available. Finally, let's focus around the challenge of regulatory compliance at early stage of clinical development. So what is the guidance provided by regulatory agencies in terms of the quality requirements for raw material? Are there any? Well, in the European guidelines for the GMP manufacturing of ATMP, it is highlighted that the quality of raw material is a key factor to consider. It is also written that particular attention should be paid to minimize the variability of the starting and raw material. And finally, that raw materials should be of pharmaceutical grade if possible, with the idea that in some cases only material of research grades are available. Meeting regulatory requirements is all about mitigating risks. By using GMP compliant raw material, you will decrease the risk on the raw material supply chain. Our GMP grade transfection reagents are manufactured through a documented manufacturing process in compliance with GMP guidelines Part 2 and Annex 1, guaranteeing the highest quality grade available. They are released with validated QC according to European Pharmacopoeia, assessing the 4 pillars of GMP compliance, namely identity, activity, purity, and safety. The regulatory requirements are becoming more stringent every day, and it has now become critical to use GMP compliant raw material to secure ATMP manufacturing. In addition to securing quality, it is also required to secure the supply of raw material. This can be done first by qualifying your material supplier as required by GMP guidelines. The qualification and approval of these suppliers should be documented, and quality agreement should be in place with key suppliers. An important tool to secure the supply process I would like to emphasize is forecasting. Performing a precise forecast of the amount of raw material needed helps to reduce the risk related to the shortage of material. This can be easily framed by signing supply agreement with your key supplier. We think that it is also quite important to build as early as possible a strong and long-term relationship with key suppliers to have them on board when moving on to commercialization. At Polyplus-transfection, we have a strong record of successful audits. We are currently investing heavily to increase our regulatory and quality assurance team and to build a new facility in order to support the worldwide demand of transfection region for virus manufacturing. And we will move this summer actually to this new facility called Vectora. In conclusion, AAV and lentivirus are the most commonly used viral vectors in gene and cell therapy, and there is an increased need for the quality and sustainable supply of raw material for virus production. We think that our range of products with Vector V AAV and PI Pro are addressing the key challenges of large-scale manufacturing of clinical and commercial grade viral vectors due to their high efficiency, versatility, and regulatory compliance. Finally, Polyplus-transfection is strongly involved in supporting the gene and cell therapy field and is dedicated to support your project. Thus, we are investing both in a new facility and in human resources, and to increase production capability, but also to develop more innovation so that we can answer your need. And finally, I would like to thank a great team at Polyplus-transfection for allowing me to putting this presentation together and for supporting the future of cell and gene therapy.